we've gotten through all of this stuff on cell walls. Gram staining, that's something that you've already been exposed to. I don't think I need to belabor that. We've got gram-positive and gram-negative organisms. Remember, the gram-positives will appear purple. Primary stain we use is crystal violet. They retain that because they have lots of peptidoglycan and no lipid-containing outer membrane. So the decolorizing step using the solvent, 95% ethanol, would normally dissolve the lipid. That's not going to be effective at removing the crystal violet. So they'll retain that color. The gram-negatives have an outer membrane which does contain lipid, the lipopolysaccharide. And so after we apply the primary stain, even though we apply a mordant, when we apply the 95% ethanol, that lipid is dissolved. It carries away with it the crystal violet. And so the counter stain safranin is what you're going to wind up seeing if you've done the stain procedure properly. And so the gram-negative organisms are going to be reddish or pink, some other color than purple. And those are the two major groups of bacteria based on cell wall characteristics. And from there, you can go further with additional, more specific tests to eventually determine the identification of the organism. This is really an important step, though, in that procedure that has to be done near the beginning. So there it is, lays it all out. All right, so one of the things that I demand that you know, if you're in a microbiology class, by the time you leave the class, you need to know the difference between gram-negative and gram-positive organisms, both in terms of their cellular structure, their cell wall structure in particular, and what they look like after gram staining. So there's all of that. And then I got through the stuff on non-typical cell walls, mentioned that we've got these forms that are gram-positives, technically, but which also contain the wax mycolic acid, the so-called cord factor. So the Mycobacterium and Nocardia, those two genera contain forms that are described as being acid-fast. There's that term, they're acid-fast. And so they will appear sort of hot pink, kind of a fuchsia color as a result of staining with carbofuchsin. I know I talked about that last time, so I'm not going to rewrite that up here. The counter stain with that one is methylene blue, by the way. So, and then we have the forms such as the mycoplasmas, which are pleomorphic because they don't really have cell walls like most other bacteria. There's a few other forms similar to mycoplasma whose cell walls are largely just sterile, so cholesterol is the main one of those. And this stuff is not terribly rigid and doesn't give a definite sort of shape, but the shapes are highly variable. So the term for that is pleomorphic. And so there are a number of forms that are pleomorphic. They may have rod-shaped cells, but occasionally you'll see them as coccus or other unusual forms, maybe Ys and little commas and all kinds of bizarre shapes. And then now we go from the cell wall inward to the cell membrane. Okay, so cell wall is always outermost, and the cell membrane is just inside of that. And as far as cell membrane structure goes, there's really not much difference between one group of organisms and the next. In fact, your cell walls, or cell membranes rather, are similar to those of the bacteria and other organisms. They are composed of this bilayer of little, why that's doing that lately. This little bilayer in here of phospholipids. So a phospholipid molecule is usually symbolized like this. And so what we've got here in this head is some phosphate. I'll just put a P there. Actually, I can put the rest of it. It's PO4, and that has two negative charges associated with it. And there's some other organic kinds of functional groups associated with that, but we refer to that as the phosphate-containing head. And phosphates, when they're soluble, when they're in water, will take on negative charge. So that's anionic and is hydrophilic. The term for that id there is hydrophilic. HYDROPHILIC, meaning that it will interact favorably with water. Water and phosphate get along. And so that's why it's facing out, because in this whole outside environment here, the interstitial region between cells, if you will, outside of the cell, there's lots of water. And so it stands to reason that it's going to face that direction. And then this end down here, I'll just label that with an FA to indicate that those are fatty acids, which are largely just carbon and hydrogen, little else there. And so that doesn't have a charge. And instead of being hydrophilic, we would say that end is hydrophobic. And notice how the tails of these phospholipids are sort of in here, sandwiched between the phosphate-containing heads. And so that region doesn't have water. It's sort of squeezed out of that space, and thus would be ideal for these fatty acids to kind of take up residence there. And so that's the basic sort of structure of the cell membrane, and for many years that was thought to be the structure of the cell membrane, but work by Dafson and Danieli and some other folks back in the 60s and through the 70s revealed that instead of just a pure bilayer, we do have a mosaic of different substances, different moieties here. And so it's a hodgepodge of things. And so we have these proteins, some of which have little dimples on their surface, as is the case here in this one. That serves as a receptor, a binding site for hormones, neurotransmitters, other messengers that are released by different cells elsewhere. And when they pass from those cells to this one can receive those substances, and it can then respond. And so cell-to-cell communication can happen. In bacteria, there are chemical cues that some cells give off to others to kind of control what they do. You've got some bacteria living in one place, and that site is favorable for their existence. They can send out chemicals saying, hey, it's good over here. We've got lots of food, or the temperature's great, or there's something about this area that is favorable. And so when those chemicals move through the environment and are picked up by binding sites on receptor proteins like this, that may direct their movements to go to the nice place. And so you can wind up with a whole community of bacteria forming as a result of that. That's in fact how biofilms sometimes form. But there are receptor proteins like that, and they're described as integral because they're inside of the membrane. They're kind of embedded in there. And then you have proteins like this one that's cut away to show that the interior of that is hollow. And so this is a space through which things can come and go. So pumps, if you will, exist in the membranes, and they can be used to bring in ions. Generally, sodium, for example, is passed out through a carrier type of protein, sort of like this one, that is referred to as the sodium-potassium pump. So we have sodium going out and potassium coming in through transport proteins like this. And then another type of protein is one that's likewise integral, but here you can see how the upper surface of that protein, that sort of blue-green part, is sticking up out of the phospholipid bilayer, and attached to the top of it is the chain of sugar. All right, these little guys here are sugars, monomers of sugar that are hooked together in series to make these little complex structures that are identifiers of that cell. They serve as sort of cues or fingerprints, if you will, that identify that cell as being a particular kind. So a particular species of bacteria might have unique sugars attached to those proteins, and that might be a cue to other members of that particular population that, hey, you know, this is one of our kind here, and so they would tend to um, uh, remain together in some sort of communal fashion. So you can have proteins with sugars attached. You can also have sugars attached, as is the case here, directly to the phospholipids. And so that combination of phospholipid plus sugar is called a glycolipid. And you've got actin filaments running below the cell membrane. You've got proteins like these that are filamentous that are extending out from the cell. So lots of different things there. And these tend to move around. Membranes are not static, but think of these as like pieces of driftwood and other items floating on an ocean. And from one moment to the next, they're liable to move to different places. And that's true of materials in this phospholipid bilayer. They can move from one place to another. Things can be taken out, things can be added. So it very much is a fluid, in a literal sense, a fluid kind of structure. All right, so cell membranes are incredibly important. And if you get nothing else from this description here of the cell membrane as being selectively permeable is probably the most significant one of the descriptions of it, because it is able to regulate what comes and goes from the cytoplasm into and out of the cytoplasm. And so as a result of that, the interior of the cell is controlled and is going to be qualitatively different from what you have in the surroundings, right? There's no equilibrium such that the outside is the same as the inside. The cell literally controls its internal environment to provide the conditions that are going to be optimal for that cell to function. If this whole thing fails and this membrane starts not to do its job, then there will be an equilibrium. The stuff that's on the outside will re-establish, concentration, within and without the cell, and you'll wind up with conditions inside the cell being virtually the same as they are outside. So cells kind of control their internal milieu. That's a word that refers to the environment inside of the cell using this cell membrane. All right, so there's that. And then everything inside of the cell is the cytoplasm, all right, which is mostly water, but other things suspended in it. So there are no membrane-bound organelles, but you have lots of different chemicals, sugars, amino acids, salts. There's lots of protein in there. I don't know why that's not included. But yes, soluble protein is abundant inside of the cell. You have ribosomes in there, lots of water. You have chromosomes and plasmids. I think I've covered some of this stuff before. We refer to this chromosome as a nucleoid, meaning that it's like a nucleus, but not really. What's not nucleus-like about it is the fact that there is no membrane surrounding this massive DNA. It's just DNA wadded up and shoved into the center of the cell, and it is always circular. And so it's like a donut as opposed to a string that has two ends. But, as far as DNA goes, it's the standard sort of DNA that we would expect in other organisms. It's double-stranded. It's composed of adenine, cytosine, thymine, and guanine. And there are segments of it that literally code for important enzymes that control functions of the cell. And so it in a big way is what is absolutely required to make that cell work. All right, so it's the headquarters of operations for the cell would be that piece of DNA. And then you've got your plasmids, which are little tiny circles of DNA that are really not essential. So they're not absolutely required. If the cell lost its plasmid, it's still going to be able to get by. The plasmids add a little bit of, additional function beyond what's normally required to keep the cell going. And so antibiotic resistance and other functions can be provided by the genes that are carried on those plasmids. And then we have bacterial ribosomes. I remember talking about those. So those are going to be free floating in the cytoplasm. We talked about how their weight is determined. These Spedberg units, these S's here refer to Spedbergs. Well, that's a measure of their ability to settle out at a particular place in the sucrose gradient. So if you place them into a test tube or culture tube containing some syrupy solution that's thin at the top and thick at the bottom, and then you spin that, say this is the tube, you put your ribosome in here, and if you put that in a centrifuge, it'll sink to a certain depth. And whatever level that is, after a set amount of centrifugation, will correspond to the weight. So heavy things sink way to the bottom, they go all the way down in here. They'll have high Spedberg values, and then lighter things will settle out higher up, and so they'll have lower Spedberg values. So the full ribosome is composed of two parts, a small subunit and a big subunit, and those together add up. They don't sum together exactly, but the weight that you get when you put the small and the big together is 70S. Right? And that's typical of prokaryotes. They have a 70S ribosome. The eukaryotic ribosome that you have in your cells, which you have lots of in your cells, would weigh 80S. Are there any 70S ribosomes in your cells? Is an important question to ask. And the answer to that is yes. And so you might be wondering, where would those be? Well, you have organelles, important energy producing organelles in your cells, the mitochondria, which have a prokaryotic origin. Those are literally prokaryotic cells that are serving as organelles in your cells. And so they have 70S ribosomes. And what's more, they have nucleoids. Your mitochondria have nucleoids and other bacterial characteristics. So, yeah, you have these, and to some extent, you're dependent on them. If you're dependent on your mitochondria, then you're dependent on their ribosomes. So, there's that. Yeah, I did talk about this. So, inclusions and granules are bodies that contain materials needed by the cell for the future. So usually limiting kinds of nutrients are stored away in these kinds of structures. Phosphate would be one of them. Iron, sulfur, some other things like that may be stored away in granules, especially when those materials are limited in the environment. It's a good sort of strategy to retain those things so that times get rough later, you can make use of them. So these are not membrane bound, and so would not qualify as membrane bound organelles. They are just masses of material that would be stored away. Even lipids can be stored, by the way. Some organisms do that because lipids have low density, and they can thus float with those. So in addition to providing a store of energy, flotation is afforded by those. And if you're photosynthetic and you live in water, it's best to ride high in the water because you can get enough light for photosynthesis. And then we have cytoskeletal elements. These are proteins that give the cell its shape. All right. And we come to the topic of endospores. And I've mentioned this previously. We actually did an endospore stain. And there was a quiz about that. This is the stain that requires use of heat as a mordant. The Schaefer-Fulton method. We apply heat and we drive the stain in because these endospores have very thick coats that are difficult to penetrate with stain ingredients, dyes, without the use of heat. So the kinds of organisms that are able to produce these endospores are always going to be gram-positive, and they are rods. Okay. The fancy word for a rod is bacillus. That's a term that describes that morphology, rod-shaped. And there actually is a genus Bacillus, so named because they are the prototypical, they're the classic rod-shaped bacteria. These other genera here, Clostridium and Sporosarcina, among others, are also all rod-shaped, and they are capable, and they're gram-positive, and they're capable of undergoing changes to become endospores. Right? And that process of becoming an endospore is called sporulation. Here it is down here, the formation of endospores. When when everything's hunky-dory and nutrients are plentiful and temperature's great and everything's fine. And this is typically early on in the growth of a population. There's relatively few individuals. They're not competing. Everybody's got plenty. Everybody is, for the most part, going to be described as being vegetative. That's the word we use when we describe bacteria that are fully active. They're metabolizing. They're reproducing. They're doing great. Okay, so those are vegetative cells. At some point, though, population sizes get large enough that they start to compete with one another. The individuals taking up all the nutrients impose challenges on their fellow population members. There's limited nutrients. There's wastes being produced. Those start to foul the environment. You can have other challenges like temperature changes and changes in the pH of the surroundings, and changes in oxygen levels may affect some organisms. Anytime, for whatever reason, conditions get tough, when there's challenges, sporulation is typically what's going to happen. And so you wind up with endospores as a result, all right? And so these endospores are incredibly resistant. The things that would normally kill a vegetative cell aren't going to affect endospores for the most part. You have to use extreme measures to kill an endospore generally, right? And so the upshot, the goal basically is to come up with a life stage that is incredibly resistant so that it can survive. Endospores are about survival. Ultimately, if they weather the challenging conditions and good conditions return, they may then reproduce. But that's secondary to the importance of these as a way to survive. Okay? So endospores represent an approach, an adaptation to survival under challenging conditions. And as far as we know, as far as living things are concerned, these are the toughest. They're the hardiest of all life forms. There may be some microbes, the prions in particular, that are not truly living, that may be a little bit more resistant even than endospores. All right, there's good indication that that's a possibility. So I'll just tell you, that's pretty impressive, because endospores are very, very, very resistant. We have to use extremes of things like heat. So we have to raise temperatures up hundreds and hundreds of degrees to literally burn these things to ash to kill them. Or if we're using moist heat, as is the case in a pressure cooker, we have to really crank that pressure up and take the temperature up well past the boiling point of water, and that'll kill them. And anything short of that won't. You can dry them out all you want, and generally that's not going to kill them. So they can withstand that usually. Freezing generally won't kill them. But radiation, yeah, radiation will knock them out if you apply it for long enough and if it's powerful enough. So gamma rays and really intense long-term UV radiation can kill them. And there are a variety of chemicals for sure that will kill them. You have some sporicidal kinds of chemicals like really concentrated hydrogen peroxide. Glutaraldehyde is one. There's a few things that are definitely sporicidal that will kill spores. And if that's the case, if you kill spores, I call them spores. If you kill endospores, if you wipe them out, if you kill them, you've killed everything else. Right? You can assume that there's no life left. And so that is the very definition of sterilization. Death of endospores. If you've killed the endospores, you've killed everything else. That's horrible, I know. Death of endospores. All right. So, and once again, I'm going to say it again. You probably have a question on an exam about this. Forming endospores is not an adaptation that is used in reproducing. This is not for reproduction. Endospores, the formation of endospores, sporulation is for survival. It's about survival. Now, once the bad conditions have passed, those endospores can actually come back out of that form and revert back to being vegetative. They'll change morphology, they'll become really active, they'll lose the coat that was protective. And so that process of returning to vegetative growth, you should know, is germination. So we've got germination and we've got, We've got germination and we've got sporulation. Right, so moving. Hopefully that turned off those subtitle things that I didn't want. But this is the sporulation cycle. All right, you can see how that works. So here's our standard vegetative cell up here. There's that guy at 12 o'clock. You can see it's got its nucleoid in there. It's got the standard cell membrane, cell wall. But then some challenge occurs, and it doesn't indicate what that challenge would be, but it could be high levels of heat, it could be competition, crowding, who knows. Something has stressed this guy out, and so its default is to just punt and say, forget about it, I'm going to stop trying to be vegetative, and I'm going to go into this endospore form. So I'm going to go through sporulation. And so they have to copy the chromosome and then sort of sequester part of that and the part that's critical, the part that contains genes that are essential, and they move it into this what's called a forespore, right? And you can see the cell membrane and some cell wall material start to kind of partition things off in the cell. And so you see this starting to grow here. There's like this little capsule that's forming inside of the cell. This part over here, I'll just draw an X through it because that's going to get jettisoned. That's going to be, that DNA is no longer going to be used, but this is the good stuff in here. All right, and so more cell wall, more cell membrane, and other constituents are going to come in there to start to surround this. And so that sac that's forming there is called a sporangium. So the endospore at this early stage is going to contain a sporangium, which literally means spore sac. All right. And additional materials will be added. An outer coating called a cortex is going to be formed that contains a variety of proteins and other very resistant, very tough substances. All right. And then a spore coat is laid down on top of that. And then finally an exosporium. So you've got 3 layers of material. The core is where the genetic material is, but outside of that, the cortex, a spore coat, and an exosporium. Once you've got that, you have a fully formed endospore, right? And that can, just basically lay there dormant. If the situation that it's in, say, just dries out altogether, let's say it's in the bottom of a pond or on the surface of a dead animal or wherever it happens in the soil somewhere, it's none the worse for wear because of that. It'll just stay there, and unless radiation or some other agent acts on it to kill it, to destroy it, it could reside there for centuries, even millennia, even for tens of millennia. A millennium is 1,000 years. There have been endospores that have been recovered from guts of fossilized insects that were millions of years old. And those endospores, when given the right conditions, when they were dissected out of those fossil honeybee guts and other things like that, when they were given the proper conditions, they re-entered the vegetative cycle after millions of years of lying quiescent and dormant. So that's, if that's not evidence for the hardiness, the ability of these guys to resist destruction, I don't know what to tell you. That's about as tough as it gets. So yeah, this describes they're verging on immortality. I mean, nothing is truly immortal, but 250 million years ago, that to a great extent predates the origin of dinosaurs. So you can have endospores that were produced, sporulation happened with them well before dinosaurs existed, and then they laid there dormant in fossil form, or in a fossil organism, preserved through the reign of the dinosaurs, and then after the dinosaurs went extinct, they continue to persist. That may seem like a really challenging thing to believe, but it in fact has been demonstrated. So yeah, this is about the only condition that normally in a laboratory we would assume has destroyed them. So in a laboratory setting, we're going to use a device known as an autoclave. I don't know that I've ever talked, I guess I have talked about them, but it's basically a glorified pressure cooker. It's a big thing that's got a steel jacket that's really tough, and you can generate high pressures in it, high temperatures with moist heat, and kill endospores. And as I said, if you kill endospores, you can assume everything else is dead, and so that's how we achieve sterilization, right? If you've not autoclaved something or treated it with an effective sterilant, a chemical or radiation to kill everything, then we can't use the word sterile, right? So if you were to take some item, say a baby's pacifier, or the little nipple that goes on the bottle that the baby would drink from. Sometimes we put those in boiling water and use different methods to clean them and say, oh, it's sterile now, we can let them have it, and she's not going to get sick. Well, the truth of the matter is it's not really truly sterile, right? It may be largely degermed, but there could be some spores there. All right, yeah, storage of phosphate. What are those? That would be granules. Now I need to talk about this. So the discussion of morphology comes up. So when we're looking at bacteria, especially when we're working with something that we're trying to figure out what it is, we're trying to ID it. Um, One of the things we want to determine is morphology. So morphology, generally speaking, refers to shape. I guess you could also say that size factors and both of those things go into determining the morphology of the organisms. When I use that word morphology, when anybody uses it, that's what they're talking about. Arrangement is also important. This is something that you're going to see at the microscopic level as well when you stain organisms to be able to see them better. Arrangements are something you're going to look for as well. Morphology and arrangement, all right? They're different things, but they're related. And there are three basic morphologies then. So one is the coccus. By the way, the plural on that, we're just going to take the US off and put an I on there. And some people pronounce that cocci. Some people pronounce it cocci. I've always said cocci because I think my microbiology professor and lab instructors long ago said that. So there are quite a few of them that say that. But they're spheres. They're just little. They're circular, but you know they're round, right? They're like a little ball, and so that's the coccus morphology. They're not longer than they are wide. You know, the same dimensions all the way around. And there's variations on that. Sometimes you have organisms that have a little bit of, say, a teardrop shape to them, sort of oblong in one direction. That still would be regarded typically as a coccus. Rods, on the other hand, the technical term for a rod is bacillus. And again, we can pluralize that one by taking the US off and putting on an I. That would be pronounced bacilli, right? Those have an elongated appearance, a capsule-shaped appearance. Some species are little short rods, some are longer. You know, it just depends. Like, for example, Coccobacillus, that's a genus. They have little short, plump rods, but they are nonetheless a little longer than they are wide, so technically they're bacillus. There's a genus known as Vibrio, which are technically rods, but instead of them being straight, they're going to have a little bit of a curvature to them. So something that's sort of boomerang shaped or comma shaped would be like that. So there's a little variation among the bacilli as well. And then a third type of morphology is the spirillum, or corkscrew shaped bacterium. Helical is the fancy word for that, but corkscrew shaped. So they're going to be sort of spiral. All right, different variants of that. Spirochetes are really long and skinny, and they tend to be built like a spring. Others are going to be more just like sort of twisty-turny. Anytime you have anything like that, an elongated cell that has some convolution in it, sort of twisted, that's a spirillum. So those are the three classic morphologies. There are, as I said earlier, some organisms that really defy pigeonholing in one of these categories or another because they're pleomorphic, so they can change form over time for a variety of reasons. But you need to know those morphologies, all right? And the good thing is often the name of the organism will give a hint about what the morphology is even before you look at it. So, for example, we have the genus Streptococcus. Right. So the root coccus is in there. By the way, since this is a group of related organisms, and that is a name that we apply to that group, it's a genus, I'm going to underline that word. Or if I have a word processor and I can italicize it, I would do that. We always set scientific names off with underlying or italicization. But yes, Streptococcus, you can see in there right away, you know that morphology, that organism is coccus. What about this one? There's a genus of organisms known as the lactobacillus. You eat yogurt, you've ingested lactobacilli. You got the bacillus root. Hey, what are they shaped like? Well, they're going to be rods, all right? So those names often are implemented when we name the organisms to highlight this important feature of their appearance, their shape. So here we've got some examples. And as I mentioned earlier, arrangement is another thing. So this picture right here shows some purple stained bacteria. They'd be gram stained, and thus they're purple. That would indicate that they're gram positive. And you can see their cocci there. And as they divide, they tend not to, spread out and become independent, but they stay kind of attached. And so they look to me like little berries, little randomly shaped clusters of cells like little berries. And so random clustering, or they look like clumps of grapes, I guess. That's the arrangement, all right? Coccus is the morphology, but the arrangement in this case is described as staphylo, right? And there's a genus of bacteria, the Staphylococcus aureus and others. That name actually tells us the shape, the morphology, and the arrangement, all in one. In fact, this one back here that I use, Streptococcus, we've got the morphology. What is strepto? Well, strepto is an arrangement. In this case, what we're talking about would be cocci, not in random clumps, but in chains. All right, so that's the strepto arrangement. Strepto means chain-like, staphylo means clumpy, clusters. So there's that. Then you got your bacilli, which also have arrangements. Sometimes they're in a strepto arrangement, sometimes they're singles. Sometimes with the bacilli, you'll see pairs hooked end to end like that. That arrangement would be diplo. Sometimes cocci are like that. You'll see them in little pairs. That would be a diplo arrangement as well. But yeah, here's some vibrios, which, as I said, typically are described also. They're rods, but more specifically, they're comma-shaped or they're sort of bent, and so we're going. Here are some corkscrew-shaped bacteria, spirochetes, and spirilla. And then you get these guys that are just all over the place, that are branching filaments. There's no fancy word for that. They're just elongated cells that branch and occasionally will give off little spore-shaped reproductive structures like this. So there's that's a potential form that they can take. And then, as I said previously, pleomorphic bacteria exist where their shapes are not set. They can be cocci, they can be rods, they can be all different sorts of shapes. So you can see in this picture here, they've circled. These are all the same species. This is a population, a part of a population of organisms in the genus Corynebacterium. I don't know why I'm writing this out, but this is the genus that contains the organism that causes diphtheria. Yay! A really bad upper respiratory infection that luckily now we have DPT immunization, diphtheria, pertussis, tetanus, and the D in there is, there's a component that helps to bring about immunization of an individual organism. And you can see they've circled a few of the cells here so that you can compare them. And obviously there's a lot of variety in this particular organism. Some of them look like little drumsticks. Some of them look like little propellers. like an airplane propeller that's got two sides to it. Some of them are more kind of rod shaped. Some of them are more like cock's eye. So a lot of variation there. So definitely these guys are pleomorphic. And then here's more on arrangement. So we got the strepto arrangement, chains. You can have bacilli exhibiting a similar kind of feature as well. You got the diplo arrangement. Cocci are notorious for doing that. Here's another arrangement we commonly see, especially with cocci, groups of 4. And this illustrates how exactly you get that. That group of 4, or tetrad as it's called, that's a tetrad, arises when you get a single cell and it divides down the middle, right? And that'll give us diplo arrangement. Then those 2 cells divide, at right angles to the first division, or they'll divide this way. Sorry, they'll divide at right angles, and you'll get something like this. And while organisms that exhibit this won't all be in tetradal form, sometimes the tetrads break apart, and you get singles and you get doubles. You'll see a significant number of these, a large enough number of those, that you can spot it. You can tell that organism has tetradal arrangement in many cases. And then a variation on that is, instead of a group of 4 laid out in 2 dimensions, you'll have another group of 4 behind that, arising again through division of these cells, and they stay attached. This little cube-like packet here, right, that has 6 faces, the cube, is called the Sarcina arrangement. And you can have those containing 8, 16, on up to 64 cells. So cube-like arrangements. We don't see those that often, but there are some Sarcina kinds of organisms that typically display that. So you can see, and then irregular clusters, that's the Staphylo arrangement. Groups of 4 are tetrads. You got the diplo. You got strepto. That's chains. And then you got the Sarcina arrangement. So those are different arrangements. And cocci, you can see, are quite versatile with regard to the variety of arrangements that they have. Bacilli are more limited. There's diplo. Some species are diplo. They commonly have 2 rods attached to one another. They can be in a strepto arrangement where there's long chains attached. And then this palisades arrangement, this is where the cells run out of space here, apparently. This is where you get the rods, and they're kind of laid side by side like this, with their ends touching. So it looks like they've kind of been compressed together in sort of like an accordion type of pattern. So you can see they're all pretty much oriented straight up and down. That's the palisades arrangement. That's one you can expect to see with some bacilli. All right, so morphologies and arrangements. Those are when we're doing inspection of bacteria after we've stained them with an appropriate technique. When we look at them with the microscope, that's one of the things we want to determine, is the morphology and the arrangement. Very helpful in identification. All right, so I'm going to go ahead and while this remaining material in here is kind of neat, I think I need to move ahead and start the eukaryotes. So this section of it, I'm probably not going to have much on this on the exam. So I'm cutting off here at slide number 44 in the prokaryotes. Okay, just for the sake of time, I'm going to stop there. So chapter 4 up through slide 44, that's where I'm cutting off. All right. So make sure you're watching these and reviewing these. If somebody you know isn't watching these, well, there's a good chance that they're going to struggle on the exam. So anyway, treat this like class, an on-the-ground class, and go through all these videos. All right, so I've got this. Now, the videos that I have for the eukaryote chapter, those are already done. Did them this spring, and so I'm going to post those as well. And so Friday for sure, we're going to, be in a position to do the exam. Whether or not I post it Friday evening or Saturday, I'm going to let you know exactly what my plan is. We'll cross that bridge when we come to it. But anyway, I'll announce that here soon. All right, so I'm going to stop it right there, and I'll be posting this as well as the eukaryote lectures this evening. All right, thanks.